Assalamu alaikum friends. In today's video you have to put patience because today the video is very much tricky, but if you watch the full video then I think, or I may say I guarantee that the genome replication will be understood by all of you guys. So you have to put patience regarding this video. I will start the genome replication by first talking about some important points regarding the genome replication, and you have to remember those points. The first point is that the DNA viruses replicate in the nucleus, while the second point is that the RNA viruses replicate in the cytoplasm. You have to remember this. DNA viruses replicate in the nucleus, RNA viruses replicate in the cytoplasm. For that I have just put two words over here, DON and CR. If for example in case it forgets from you guys, then DON D stands for, or D represents the DNA and N represents the nucleus, while in CRC represent the cytoplasm and R represent the RNA. So RNA viruses replicate in the cytoplasm and DNA viruses replicate in the nucleus, right? Okay. Now DNA viruses replicate in the nucleus. From this we conclude that DNA viruses will use host enzymes. Okay, this is the other point. DNA viruses will use the host enzymes because they replicate in the nucleus, and in case of RNA viruses, as they replicate in the cytoplasm, so they will use, or sorry, they will carry their own enzyme for replication. So this is also important. Okay, moving onwards, we know, as we are studying the genome replication, and we know in replication there are two processes. One is the transcription, the other is the translation. Transcription is the formation of mRNA from DNA, right? So here we are going to synthesize the mRNA. So how we are going to synthesize mRNA? For that we will use a strand, and that strand will be antisense strand, or you may say the negative polarity strand, right? Now from that, if when we synthesize the mRNA, then from mRNA we will synthesize the protein, and for protein the template required will be negative polarity, right? Or you may say antisense template. So here what I get the point that for mRNA the template used will be negative, while for protein synthesis the template used will be having positive polarity. Okay, now moving onwards, these all are important for your exam point of view. Okay, so keep patience. Now when there is conversion, when we are or formation of RNA from DNA, so we are making RNA from DNA. So the enzyme name will be DNA dependent RNA polymerase, or you may say RNA polymerase, right? Okay. Now if we are going to synthesize the DNA, right, from RNA, so the enzyme name will be RNA dependent DNA polymerase. Similarly, DNA dependent DNA polymerase, RNA dependent RNA polymerase, right? So this was also important. Okay, now here we are moving onwards towards exceptions. I told you that all the DNA viruses replicate in the nucleus, and the exception is the pox virus. The pox virus replicate in the cytoplasm. We studied that all the DNA viruses replicate in the nucleus. The exception is pox virus. Pox virus replicate in the cytoplasm. Okay, moving onwards, we said that RNA viruses replicate in the cytoplasm. The exception is here. What are the exceptions? The exceptions are the retroviruses and the influenza viruses. Both these viruses replicate in the nucleus. Once again I will repeat, the RNA viruses replicate in the cytoplasm. Exception is there, and that is retroviruses and influenza viruses, they replicate in the nucleus. Okay. Now moving onwards, most of the DNA viruses are double-stranded DNA viruses, except parvovirus, that is single-stranded DNA viruses. Now most of the RNA viruses are single-stranded RNA viruses. Exception is there, and that is reoviruses. So reovirus is the only virus, is the only double-stranded RNA virus. Parvovirus is the only single-stranded DNA virus, and retroviruses and influenza viruses are the only RNA viruses that replicate in the nucleus, while the pox virus is the only DNA virus that replicate in the cytoplasm. The rest of all DNA viruses replicate in the nucleus, the rest of all RNA viruses replicate in the cytoplasm, and the rest, most of the DNA viruses are double stranded, most of the RNA viruses are single stranded. So this was all important regarding the MCQ's point of view and regarding the topic that we are going to discuss. We are going to discuss the genome replication. Okay, now this is our main topic of discussion. Okay, so we have genome, a DNA genome. So it may be single stranded DNA genome in case of, as we study single stranded DNA virus. Here is parvovirus is the example. It's single stranded DNA virus, right? So for example, if we have parvovirus and that is single stranded DNA genome, that has single stranded DNA genome, so what we are going to do, we will require, okay, this single stranded DNA genome, maybe it may be positive or it may be negative. So first we are going to convert it into double stranded DNA genome, as this is DNA virus. So I told you, if the virus is DNA will replicate in the nucleus, and from that we conclude that it will use the host enzymes. So this will use the host DNA polymerase, right? So the single stranded DNA genome will use the host DNA polymerase enzyme, and it will be converted into double stranded DNA genome, right? Now if for example it is negative strand, it will be converted into positive, means the other will be positive. If it is positive, the other strand will be negative. But in both cases, the answer or the product will be double stranded. We will the end product will be double stranded DNA genome, right? Okay, so we have here the positive strand and the negative strand, right? Okay, once again I will repeat. I have the enzyme, I have the genome, single stranded DNA genome, and this is of parvovirus, right? Parvovirus is the only DNA virus that has single stranded DNA genome, right? Okay, parvovirus. Now we need to produce the viral mRNA for the viral replication, right? Because we have to synthesize protein, and the protein must be synthesized through transcription and translation. Okay, so first in transcription we are going to synthesize the mRNA. Okay, now here we have two strands, one is the positive and the other one is negative. We discussed that for synthesis of mRNA we will require an antisense strand, right? And the antisense strand is also called as the negative polarity strand. So what will happen, this is the complementary of this, so we will synthesize the mRNA, right? So the mRNA will be synthesized by using what? By using RNA polymerase of the host enzyme. Now why I'm going to say RNA polymerase? Because it's the formation of RNA from DNA, right? Double stranded DNA is converted into RNA. So the enzyme here will be DNA dependent RNA polymerase, right? So in short, I can say the product we get is the mRNA, and this mRNA is basically the viral mRNA strand, and it will be further processed and moving onwards for the translation process. Okay, now if we have double stranded DNA genome, right? For example, we have this double stranded DNA genome. Simply by using DNA dependent RNA polymerase of this is the host enzyme, right? Because this is the DNA virus. Okay, so what will happen, the mRNA will be synthesized, and this mRNA will be used as viral positive mRNA strain, right? So this was all about the DNA genome. If you guys have any problem regarding this topic, you can ask freely. I'm here to answer your questions or entertain your questions. Okay, now moving onwards towards RNA genome. So we know that most of the RNA viruses are single-stranded RNA viruses, right? Okay, now we have single-stranded RNA genome. If suppose this is of positive polarity, so it will be used as viral mRNA. There will be no conversion in case if this single-stranded RNA genome is not retroviruses. But if it is retroviruses, so retroviruses basically infect human, and in order to infect human, as human genome is DNA genome, so it has to be converted first into DNA, right? So first we are going to convert this positive single-stranded RNA genome into and making DNA copy. Then that DNA will be used to integrate, or then that DNA will then integrate with the human DNA, and as human DNA replicates, the RNA genome will also replicate, and then the RNA portion, the viral portion will detach, and then it will be used to synthesize the viral mRNA, right? So in case of retroviruses, if we have positive single-stranded RNA genome but the virus is retroviruses, it means it will infect the human. So first we are going to convert to make a DNA copy. And how we are going to DNA copy, as this is RNA, so it will be synthesized in the nucleus, because this is the exception. Retroviruses, the retroviruses is the exception. Retroviruses and influenza viruses, they basically replicate in the nucleus. Okay, so what happened now, they are in the cytoplasm, the single-stranded RNA genome, they will use their own viral DNA polymerase. So when they use viral DNA polymerase, there they will form the DNA copy. Once the DNA copy is formed, the DNA copy will then enter the nucleus, and there they will integrate with the human DNA, and then through multiplication it will multiplicate. Now then when they detach, then they will be required, or they will be then converted into viral mRNA by the host RNA polymerase, right? Okay, so this was all about the in case of retroviruses. Now if we are going to talk about if the RNA is negative polarity single-stranded, so what we are going to do, we have to convert it into positive strand. For example, if you can see over here, we can directly make an mRNA copy from this single-stranded RNA genome. And how we are going to do it, by using this is RNA, and it is going to replicate in the cytoplasm. So this will be viral RNA polymerase. So by viral RNA polymerase, it will be converted into mRNA, right? As this positive is complementary to the negative. And for the synthesis of mRNA, I told you earlier that the mRNA required the antisense strand. So this is the antisense strand for mRNA, right? Okay, now moving onwards, we have, if we have double-stranded RNA genome, one will have the positive polarity, the other will have the negative polarity. So the negative polarity strand will be used as a template, and the mRNA will be formed, and that mRNA will then be used for the synthesis of protein. And the enzyme used will be as this is RNA virus, they replicate in the cytoplasm, so they will have viral RNA polymerase, right? So this was all about the RNA and DNA genome replication. If you guys still have any problem or any doubt regarding the topic, you can ask freely. I am here to entertain your answer. Thank you very much. Allah Hafiz for now.